Today, we are going deep, diving into the microscopic universe of the human egg, or the oocyte. This is a world where the success or failure of creating a new life is decided by events happening on a scale smaller than the width of a single human hair. So let's start with the big question. What actually makes one egg healthy and another one not so much? I mean, it's a huge deal, especially when you think about fertility treatments like IVF. Picking the best oocyte is always the first and maybe the most critical step. For a long time, we've been looking at the obvious parts, the nucleus, the cytoplasm. But what if the answer is hiding somewhere else entirely? Well, it turns out the key might be right here, in this incredibly active and intelligent layer just under the cell's skin. This is the oocyte cortex, and believe me, it's so much more than just a boundary. It is, for all intents and purposes, the oocyte's mission control, and it runs the entire show. To really get how important this is, try not to think of the cortex as some kind of passive wall. Instead, picture it as a bustling command center, one that's constantly working, prepping for what is arguably the most important job in all of biology. So what is it exactly? Well, the oocyte cortex is this dense, super specialized web of proteins and signaling molecules. Now, most cells have a cortex, but the oocyte's is unusually thick. We're talking several micrometers. And that thickness isn't just for support, it's because this cortex has a whole checklist of highly specialized jobs to do. And its first big job, communication. You see, an oocyte doesn't just grow up in isolation. It's surrounded by a whole team of support cells called cumulus cells. And the cortex is the bridge that connects the oocyte to its entire support system. And this slide shows you exactly how that works. You can see these little structures. They're called transzonal projections, or TZPs. Think of them like tiny cables reaching out from those cumulus cells and plugging directly into the oocyte's cortex. It's a non-stop, two-way highway for information. On one side of this highway, the oocyte is constantly receiving nutrients and really critical signals. These signals basically keep it in a state of suspended animation, a process called meiotic arrest, which pauses its development until the perfect moment. And in return, the oocyte uses its cortex to send out its own chemical messages, managing the behavior of all those cells around it. It's a vital conversation that's essential for growth. But the cortex's job goes way beyond just communication. As the oocyte gets ready for fertilization, the cortex itself goes through this incredible physical transformation. You can think of it as a really delicate mechanical dance. And you can literally see the change here. On the left, you've got the immature oocyte, and on the right, the mature one. This isn't just a surface-level change. The cortex is fundamentally rewiring its internal structure, changing its physical properties to get ready for everything that's about to happen. And the scale of this change is just wow. During this maturation process, the tension inside the cortex, basically how stiff or soft it is, drops by about 6 times. This softening makes the oocyte way more flexible, which is absolutely crucial for its next big task, getting all its genetic material into the perfect position. This whole transformation is pulled off by a team of highly specialized proteins, which you can see right here. To put it simply, some of these proteins work to create a soft spot in the cortex, while others create a stiff spot on the opposite side. This difference in tension helps build a network that literally pulls the cell's genetic machinery, that's the spindle, to the exact right spot for a healthy division. It's just a beautiful, coordinated piece of biological engineering. Now, this whole process is incredibly precise, but, because it's so complex, it also means there are plenty of chances for things to go wrong. So let's take a look at what happens when this delicate dance gets, disrupted. So what happens if this delicate dance gets out of step? What if the proteins don't do their jobs right, or if the cortex just doesn't get into the right physical state? Well, the consequences can have a direct and serious impact on whether a future embryo is viable. It turns out cortical stiffness is a real balancing act. As you can see here, it has to be just right. If the cortex is too stiff, it gets in the way of key processes. But if it gets too soft, that can lead to major errors in sorting the chromosomes. And that is a leading cause of something called aneuploidy, which is just a term for an embryo having the wrong number of chromosomes. Another big problem that can pop up is something called embryo fragmentation. This is where little membrane-covered bits of the cell just pinch off, which really lowers the quality of the developing embryo. New research suggests this might happen when the machinery in the cortex that's supposed to eject a polar body gets switched on at the wrong time or in the wrong place. It basically starts treating parts of the embryo like they're genetic trash to be thrown out.
So, this deep dive into the cortex isn't just cool science for its own sake. It's actually opening up a whole new frontier for how we assess fertility, a frontier that you can, quite literally, feel.
And this is where it all comes together. Scientists are now realizing that these physical properties, the tension, the stiffness, the elasticity of the cortex, are actually powerful signs of an oocyte's health and its potential to become a healthy embryo. They're basically powerful new biomarkers.
For instance, check this out. Studies in mice have found a direct link. Oocytes from younger, more fertile mice are significantly stiffer than those from older mice. This connects a measurable, physical trait directly to how likely that oocyte is to develop properly. And why this matters is huge. It gives us a brand new, non-invasive way to potentially grade oocytes. And what's really amazing is that embryologists are already getting a sense of this in the clinic. It happens during a very common IVF procedure called ICSI. That's when they inject a single sperm into the egg. When that tiny needle penetrates the oocyte, the way the cortex and the membrane react provides immediate, physical feedback on that egg's quality.
And they've pretty much observed three general patterns. Some oocytes show very little resistance. That's a good sign. Others are super elastic and springy. But then there's a third group that has a sudden breakage, where the membrane just breaks easily without much stretching. And that last pattern has been linked to lower survival rates and worse outcomes. The oocyte is literally telling us about its health through its physical response. And as the researchers themselves state, this opens up a very real possibility for the future. They imagine an oocyte aspiration assay, which is just a fancy way of saying a test that measures these physical properties. This kind of test could one day help us predict an embryo's chances, maybe even better than just looking at it under a microscope can. Which brings us to a really provocative final thought. From a complex symphony of proteins all the way to the simple physical reaction to a needle, we're learning that the secrets to an oocyte's potential might not be hidden in its genes, but in its very structure. Could the future of fertility really be predicted by the feel of a single cell? It's a question that brings us right to the cutting edge of science and patient care.